we've had this incredible acceleration, an exponential acceleration of being able to read, write, and edit DNA, which is giving us this incredible power to manipulate biological systems.
Hey, everybody, this is Dr. Bob Hariri with Piloting Health, and I'm very, very fortunate today to have a friend and a really noted scientist, geneticist, futurist, a friend of mine, Dr. Andrew Hessel, who's going to be with us talking about some of the unique intersections between his world, the world of genetics, and the future of genomics, and how we're going to control our destinies and the quality of our lives, as well as someone who I'd consider to be an inspiration for how we're going to take our technology and apply it to the future of preservation of performance, longevity, and who knows, maybe a form of immortality. Andrew, good to see you.
Great to see you, Bob.
Would you mind giving everybody a little bit about your background, because they're going to be interested and they're going to want to know about your book, The Genesis Machine, and as they get to know us together today, they're going to get very curious about what your future looks like.
Sure. By training, I'm a microbiologist and geneticist. I started doing genome mapping of bacteria 30 years ago almost, and absolutely adore single-celled organisms. And that work has taken me to some really interesting places. I worked for Amgen for many years. I've worked for Autodesk doing design and design of organisms. And today, my professional position is I'm the chairman and co-founder of the Genome Project Right, which aspires to be the other side of the coin to the Human Genome Project, not reading, but learning how to write large, complex genomes. So I'm basically a huge biogeek. By the way, just to be clear, I'm not a doctor. So thank you for the honorary doctorate, but I've had a weird career path, but ultimately I was hired away from my PhD program.
But in our world, you're considered at the level of doctoral level scientists. So I apologize, but that's how I always am going to refer to you internally. Um, the reality is that you and I have known each other a long time through our community. Our colleagues like Craig Ventner, Peter Diamandis, Ray Kurzweil, all of us have the same personal kind of mission, which is how do we reduce what we learned about how our cells are programmed using the coding language of DNA. How can that programming be affected in order to optimize health and potentially control our lifespan? So give me a sense of what's turning you on in the field.
What's turning me on these days is that we've had this incredible acceleration, an exponential acceleration of being able to read, write, and edit DNA, which is giving us this incredible power to manipulate biological systems. So I'm really bottom up on that. Of course, most people know that the sequencing is incredibly cheap today and very powerful. It's our microscope into living systems. DNA writing has been lagging. On a per base comparison, it's about 20 million times more expensive to write a base pair of DNA than to read it. And of course, our ability to edit DNA with tools like CRISPR, et cetera, is already moving to the clinic and changing the game in cancer and many therapeutic approaches. But it's that core technology today that's really accelerating things. And add in AI, which now allows you to assimilate a ton of data or written information, and it's just there's never been a better time to be a biologist.
You've been one of the pioneers in synthetic biology. So for the audience, tell them what synthetic biology means.
Well, no biology is synthetic. It's all based on the machinery of life that we all share. But the core technology of synthetic biology is the DNA synthesizer. It's basically a 3D printer for the DNA molecule. And what's beautiful about that is now you can do all of your editing and programming using software tools to make a digital DNA design. And when you're ready to start moving it into living systems, into the lab or into the clinic, you hit the print button and you turn that digital DNA into molecular DNA that the cellular systems can run. This is great for making a program. If you make a small program, you're going to make a protein. If you can write a longer program, you can make an entire organism.
You know, the audience probably gets excited when they hear about some of the concepts we're using where we're editing our DNA. And some people may be familiar with terms like CRISPR and enzymatic ways of kind of splicing in and splicing out segments of our DNA. But you and I have always seen it the same way, which is DNA is like a computer programming language, and that we now have the tools to change that programming, and that ultimately translates into a change in the biological performance and behavior of our cells. And this has got far-reaching implications, right? We're doing it, we're editing cells so they express a CAR construct on the surface so they can go and hunt and kill a cancer cell. But there's a lot more to it. So bring us up to speed. What's happening in the field that you think has the nearest term clinical applications?
So, oh man, that's a pretty broad spectrum. One of what's happening today that is huge is that protein engineering has gone fully digital. We now have the ability to analyze just from genetic code the form and function of a protein. We can predict it computationally, which is amazing. We can design novel proteins computationally. We can export the genetic code to make that protein. We can synthesize it from scratch, and we can express it in cell-free or cellular systems to make that protein and run it and compare it to our digital designs and simulations. That's huge. And with every step change in our ability to synthesize and assemble DNA, we keep moving up the biological hierarchy. Instead of just a protein, we can do networks of proteins, which we call metabolism, or a complete cellular genome, which is a complete self-replicating group of proteins, essentially.
And I bet you the audience doesn't know it, but we've actually been successful at creating an organism, a microscopic organism, by essentially programming the DNA. Craig, I know he got a lot of notoriety for creating that first synthetic microbe. Explain how that works.
So that first synthetic microbe was a tour de force. In all my presentations since 2010, when he did that work, I've said Craig deserves a Nobel Prize. And in fact, I wrote to the Nobel Prize committee saying every year he does not get a Nobel Prize is kind of shame on you. But he synthesized and assembled a 1 million base pair bacterial genome for a mycoplasma species, booted it up and demonstrated that it's really the first living organism where that has been engineered. He didn't create life, but he learned how to build a program that is essentially the operating system for life and boot it up, which is huge. And since then, other groups have been moving towards that space, but Craig has been the leader. And now, but I want to see many more people follow in his footsteps. It's just a technology problem at this point that our synthesizer assemblers are still too expensive for doing bacterial scale genomes or larger.
For those Star Trek fans out there, where we talk about the future of teleporting, I remember that I was with Craig that weekend where he basically took a genomic code, transmitted it digitally, and then reassembled it in a remote location, which is kind of a version of teleportation, I guess.
Yeah, people call it teleporting an organism or faxing an organism.
Faxing an organism.
It works. It's used commonly now in the virological space, where because our technology today allows us to build viruses inexpensively. When SARS-CoV-2 first appeared in China, essentially we teleported that virus, to labs in Switzerland that booted it up. And of course, it was used for vaccine development as well.
So the future is bright. Let's talk about an area that you and I both think is kind of exciting, which is we have the ability today to recover and isolate cells from any individual in the form of a stem population, which retains that differentiation versatility and that high degree of proliferative potential, so you can basically mass produce replacement cells, if you will. You and I talk about what's been done for cord blood banking for newborns, where we collect those leftover cells that are normally going to be discarded, process and cryopreserve them so they're available decades in the future in case the child needs it for a bone marrow transplant or whatnot. But as you know, at Cellularity, we've just launched an adult cell banking program where, from a simple procedure of collecting your peripheral blood, we can isolate the mononuclear cell fraction and some of the stem cells, and then have that starting material as a basis for immunotherapy. But where I'm really excited is, can these actually be used to augment or support your damaged or depleted population of cells as you age? So we know that one of the hallmarks of aging is a combination of defects to the biological software of your cells and the deficiency of those cells. So if you have a supply of starting material, you might be able to augment it in the future. So you and I both want to have our cells stored away so that we can improve the quality of our lives as we age. What else do you think we can do with these cells?
I think the future is yet to be written, but it's massive. Again, your program, your genome, is the operating system for you. It's unique. It does degrade over time because our cells don't turn off; they accumulate corruption. So the sooner you can bank your cells, the better. And we're learning, as you said, to use them in more practical ways. Whether it's to do a cell therapy on the research side, we're learning how to turn those cells into organoids and study tissues and do drug testing. But one of my favorite applications, and this is far future, is just bank yourself, because you have no idea what's going to happen 5, 10, 20 years down the road. And the one that the reason why I champion banking ourselves as adults today also is I want to see, I've written about this, it's out there. I want to be cloned after I'm dead. And it's not because I feel I'm anything special. It's just that the other options of just being buried in the ground or cremated just doesn't seem that interesting. And I read Dune when I was like 9 years old, and Duncan, Idaho was serially resurrected for 5,000 years. So it's like, so I'm not going to push cloning technology today, but my kids are IVF. It's very similar to do cloning, but I can leave that for the future. But if you bank your cells, you're prepared for that future, even if it's 100 years away.
I talk about this in small circles, that we have the ability today to take all the starting material to create a de novo organism, or to support an existing organism by simply cryopreserving at these ultra low temperatures, high quality stem populations that can be re-administered at a point in the future. And people get nervous when they hear the term cloning because they don't fully understand what's meant by it. All we're talking about here is using a starting cell to produce another population of cells that can either occupy an organ or a tissue, or under the right circumstances, create a whole new human being. And we know that it's possible. Dolly the sheep was an example of being able to clone a mammalian species. And look, there's so many efforts out there to create some pathway to immortality. Either downloading your consciousness and then uploading it into a computer, or transferring it. All these things are what our people are pursuing. But I don't see necessarily the downside in the future of being able to recreate an individual in the same biological form that they began at some future location or time.
Yeah, it's just having a time displaced twin brother or sister.
Exactly.
And so for me, it's just having another life cycle of the genome. The most complex aspect of this is it's not going to have the same parents I had or you had. So you have to think a little more downstream there. This is why it's almost like writing a science fiction story at this point, because it's not there. But I love the idea that we're going to be able to do tissue regeneration, that we're going to be able to do repairs of damaged tissue, that we're going to potentially be able to reconstruct limbs. But the idea of being able to produce another instance of ourselves, or to produce the gametes to have children even down the road, is huge. And right now, it's in the realm of the future, the possible. But these futures come faster than sometimes we're prepared for.
It's a really good point. And the fact is, immortality, as we intellectualize it, right, as we think about it, okay, comes in many shapes and sizes. immortality can be extending life beyond the predictable lifespan today, 80, 90 years. If we extend it to 130, 140, 150, are there ways to do that and give a relative immortality to people? And one of the ways is to simply look at what parts of the body, what parts of our organs and tissues degrade predictably over time, but are replaceable. Because if your brain can remain cognitively functional, but the parts of our bodies which degrade, like lungs, kidneys, joints, heart, if they can be replaced because we use these cells as starting material for organogenesis, then maybe you can get an effective preservation of performance so that lifespan can actually begin to approach a relative immortality.
I love the idea of that my organs are regenerating as I age and continuing to stay functional and young. I love that. And you know, again, we're just at the start of learning these programs and how to manipulate these cells to do what we want them to do. But the knowledge is accelerating extremely quickly. So, you know, just to take it back to the banking, I think I want to see, you know, I want to see the banking industry pay more attention to this banking sector because it's one of the few areas that we don't bank yet. We do it for some creatures. We do it professionally for some for pharmaceutical development, et cetera. We have biorepositories. We have something like the UK Biobank, which is changing our understanding of human populations because we've collected the metadata around the genomic and other functional data. But we don't have the Wells Fargo, the JP Morgan Chase yet of banking our own biology and the biology around us that we love, including our pets, our children, et cetera. It just opens so many options. Cord blood banking was ahead of its time in some ways, huge industry. I would really like to see that, you know, every person born be banked moving forward.
Couldn't agree with you more. And the tools are here, the costs have come down, and the ability. I mean, here's the bottom line. People say, how long are these cells good for? Here's my point of view. Everything we tells us today, cells you bank today are going to be good for the lifetime of the donor. And you know, we already know that cells cryopreserved 50 years ago, when thawed, are as good today as they were when they were cryopreserved. And the last thing I want to talk about is that there's also this important convergence of AI, machine learning, and our biological specialties. And so there's no doubt that the data science tools to be able to download enormous amounts of information about an individual and then re-upload that to any new form that individual takes is a way of kind of creating almost a durable memory. So I'll give you an example. I'm you know, as a guy who used to love watching specials about the way the Egyptians treated their dead, which is to preserve, as you know, they preserve them in mummification with organs isolated and stored away in the burial chamber. The life history of that individual is recorded on the walls. The thought was that at the time of resurrection, that new form that was created would be re-educated about the life of the individual, and they would essentially come back in the next generation. It's almost like the next iteration of that life. You know, we have all the tools to do that today.
I agree. You start seeing companies like Neuralink really taking brain-computer interfaces far forward. There's a possibility that, you know, in the future you have an interface and you can just literally access your digital memories with, you know, that are perfectly preserved, even better than our natural memories. But you know, I always tell people one of the areas that we have to invest more in is understanding how memory is encoded in our brains. That's a huge area for R&D that I think will have massive payoffs.
And we're fortunate that people like Elon Musk are doing really cool things to show that biology and hardware can be interconnected, and you can get a meaningful transfer of data that's bidirectional. And that I think is very, very exciting. All right, so last but not least, as a consequence of your great book, The Genesis Machine, Is your next project going to be a fiction novel, a science fiction type product?
Yeah, I think it has to be because the gap between science fiction and real world science is narrowing so quickly. And so to kind of open up some of these ideas and their possibilities, I think you do it in a fiction realm. It's a lot, it doesn't need FDA approval. So you can get the stories out there. And I think that plants the seeds for a lot of R&D and investment moving forward.
Let's face it, some of the greatest breakthroughs have occurred after an inspiration from a science fiction novel, right?
Absolutely.
You and I are both victims of that. Well, Andrew, it's great to see you, and I can't thank you enough for taking the time to talk with me. Looking forward to working with you on a couple of these projects and keeping the folks informed. But great to see you, my friend.
Thank you, Bob.
All right, everybody. This is Dr. Bob Hariri at Piloting Health. Thanks for joining us. Some of these things might have got your creative juices flowing. Continue to follow us on Piloting Health, and we'll update you on the next iterations here. Take care.